Dr. Ali is Senior Director of Scientific Affairs and Technical Marketing at Ascendia, who are actually having a booth at our exhibition area as well. He was also Technical Support Manager for BASF for many years, over 18 years, and that's where all of us have probably known him from his past. But certainly, again, I over the past 20 years or so that I know, Shaukat, I have learned a lot from him. He has. He was, as his first position, he was Assistant Principal Scientist at Liposome Company Incorporated, has a PhD from the City University of New York, and he will, again, bring us back to a topic about bioavailability and how to enhance it. With that, I welcome again Dr. Ali on the stage.
Good morning, everyone. So thanks, Prabhu, for a nice intro. Today, actually, I'm going to talk some of our technology. There are 4 different technologies that I'm going to give very highlight, overview, and then we'll take a deep dive on few technology that we are going to, you are using. So let's move on to the next one. Okay, here we go. So. All right. So just to give a brief intro about the Ascendia, what we do. As Firoz mentioned, we have a booth here in the back. And then the second, actually, bullet point, looking at the celebration technology, that we have 4 different technology, focusing on one of the, actually, maybe 1 or 2, just giving, actually, the broad overview of the 2 of the technology that mainly apply to the injectable formulations. So I'm not going to the oral or the other sets and smears, other formulations, but looking more in the nanosuspensions and also the lipid salt, which is the liposomes, lipid-based technology, or lipid nanoparticles, and then at the end we summarize. So Ascendia founded back in 2012. But Dr. Jim Wong, having worked in the industry for many years, he realized, I think it's time to start a company. So he founded this company, not far from here, actually, just next town, North Brunswick, if some are familiar with this area. It's right on Route 1 in New Jersey Bioscience Center. So easily accessible, actually, from the northeast corridor, very close to the airport as well. And we have 60,000 square feet facility, which is really the building you see with the red arrow where we are located. So everything actually done right in the building. We moved actually 4 or 5 years ago, building next to it. So we expanded, we raised the fund, and also we built our manufacturing side. So I'll come back to those. And then, then we have had a lot of talents since I came 2.5 years ago. So the company has grown tremendously, actually, right? So we are building the, phase 3 facility. Right now, we can go up to phase 1, phase 2, but we are also building for the phase 3, and also commercialization of the, drug products from outside as well. So everything is based in the U.S. It's nothing in China, because many people actually think that we are a Chinese company, but it's not actually. We are based in the US. Like I said, we have the GMP lab, both the aseptic and also non-aseptic. We call sterile and non-sterile labs. And we are growing as well. We are hiring a lot of talent. And like we have grown at least, 40, at least 20-30% since I joined 2.5 years ago. So Ascendia is a CDMO, a specialty CDMO, and I'll say why we are a specialty CDMO. We have projects, very high-end projects, because those require very special manufacturing capabilities and also the expertise that we have in-house. Focusing the formulations, mainly the more innovative drug molecules, more the novel the compounds. We have also some repurposed drug as well. For the complex generic, they are already marketed, but then people also want to develop and bring to the, clinic and go to the market, faster. So we take some of the projects as well. Particular more requires very intricate formulations, very difficult formulations, so we can solve those, challenges. How we solve this, challenges? We have the 4 different technology. We have the Emulsol, Emersol, Nanosol, Lipid Sol. These are all the, Sol means solubilization technology. Best Emersol, by name you can see is amorphous dispersions. The Emulsol emulsification system, emulsified, and we, Gattefossé and other, they spoke about the said systems yesterday. I'm not going to talk today. And the other one, Nanosol and Lipid Sol. So we're focusing actually today, going more on the injectable side, keeping, and so we'll talk about some examples, case studies of Nanosol and also the Lipid Sol. And as I said earlier, we are, expanding. We do have the, aseptic facilities that we can take it, to all the way to phase 1, phase 2, and then also building for phase 3 as well. So just coming back to the, giving the overview, we can take all the way from the, like we have, aseptic area 1, aseptic area 2. And again, these rooms are required special, actually, cleaning and all those protocols, so that we have the ISO 5, 6, 7, and we can go up to 30,000, 25 to 30,000 vials. And we can, and then we can also, building, as I said, you can go up to 150,000 by next year. So, we have several projects in the phase 2. We are building in anticipation. The project will go in the phase 3. And, so hopefully we'll have the bigger, facilities. We have also a space available in the NJ Bioscience Center next to it that we can expand, also if there need be, right? So, but we have quite a lot of space, we have both the injectable and also the oral facilities as well. So we can take not only the injectable, but also the, oral, that some of the projects requires the spray drying, early phase. We require the nanosuspensions for the oral, capsules and so forth, emulsification system, So we can do all this actually. We don't manufacture the soft gel, so we don't have, but we can take it to a hard gel capsule. The machine you see actually at the bottom, this is our colonel filler. This is actually equipped, automated, filling device, everything. We can take it from 1 ml to all the way to 20, 100 ml vials, cartridges, and also the pre-filled syringes as well. So we can, we have fill equipped. We can take it to the, to the manufacturing all the way. And then we also have the lyophilizers, because we have 2 lyophilizers, 50 liters, so we can go up to 100 liters, in the lyophilize. Because a lot of, we have formulations, the customers looking for lyophilizers, because not many places they have, and we do have in the ISO room 5 that we can, more mainly for the injectable products, right? And very high values. So we low volume, high value products that we have. That's the reason actually we say specialty CDMO. We're not like, other places. There are many places actually like us to find, because customers they find and they come to us because, they have, shopped around and eventually they come to us. So let's take a little bit, deep dive into the technology. We have 4 different platform technology. This is enabling technology, and that if customer comes up and have no clue how to handle those molecules, we can really get into the take a more holistic approach. And many of the customers, they know exactly what doses. I know everyone, whether they want to oral or, injectable or, what kind of topical. So, but here we have a more holistic approach, and we provide the, very seamless support to the customers. They are, they have no clue actually sometimes because they are virtual companies, and they really give us to us. We can use this technology. If it's tablet, we can look in the amorphous dispersions, nanosuspensions, particularly the emulsification system, then, Emulsol. But more specifically, there's a lot of interest in the parenteral. So nanosuspensions actually is a hot area now, particularly long-acting injectable, right? So if you look at the products, people have oral tablet, taking like 3-4 tablets, maybe, many tablets a day. And particularly in a HIV area, right? I mean, you can see those patients, they have to take many tablets, many pills to, keep their health. So there's a lot of interest in the antiviral areas, right? So nanosuspension actually is one of the hottest area. And that we can do it actually here in the US actually. So we have some big machines we can manufacture on the GMP. So you can see this column, this shows just giving a summary of those technology. MRSOL, it stands for amorphous dispersions. We can also design. We can look at the polymers. They are pH independent, are dependent. Sometimes we have the food effects. We can also, we can mitigate. We can look at the more holistic approach and see which of the polymers actually can be used, for particular molecules. So like if the drug is, weakly basic or weakly acid, we can look into that point of view. And nanosol is widely, accepted or used, many, not only one doses, but also multiple doses as well. So topical, ocular, and you name it, right? So this is really the area that we have several projects also for the ophthalmic, nanosuspensions, otic, in the ears, also the oral as well, right? Because we had a client, they couldn't really solve the problem by amorphous dispersions. And they came to us, because they had high pill burden. And by using this technology, able to solve the problem. We can reduce the pill burden by using nanosol technology. The emulsol is more into emulsification, so we need more surfactant. The difference between the nanosol and the emulsol, of course, we need surfactant, solubilizers, PVPs, different polymers to stabilize. As you know, we all know about amorphous dispersions and also how to stabilize those particles. Particularly, actually, when the nanosuspensions, these particles are high energy, right? And how to mitigate those and how to really cool them down, because just use the polymer, and so the interactions, entanglement with the polymer makes them more, calm down. They are more stable in that sense, okay? And the lipid is one of the latest technology. This is actually, I'm going to a little bit focus what it is. We also have some papers in the back, so if you stop by, we can read some, literature as well. So lipid cell technology, this is the actually, I drew this diagram. Really, it really takes you to all the way to the end, right? So for example, the liposomes. Liposomes have been around for a long time. I started my career with liposome back in the 80s when I was doing my PhD in City University. This is really the area at that time, actually, it was booming, actually, right? It's a lot of interest with paclitaxels, a lot of interest with the doxorubicin back in the 90s. If you remember, doxorubicin was launched in 95. That was the first drug, actually, with the liposome, right? And if you see the origin of the liposome, it started back in 1965 by Alan Bungham, right? So it took almost 30 years, plus years to bring this, one product, doxorubicin, in the making, in the market, right? And then, followed several other drugs. But it's still, there are a handful of products in liposome. And the reason being drug loading, number one, and also the stability, right? The liposome, they start leaking, and also, that really creates a lot of problems. So often you tend to lyophilize, right? So lyophilization becomes a media to able to lyophilize, to stabilize the liposome. With the cryoprotectant, you can use, mannitol, other sugars, we can do that. So this really gives you a snapshot. The liposomes they're the good one, because then you can also functionalize those liposomes, depending what organ you're targeting, right? So if you have immunogenic liposomes, you can use some, antibodies. We can also use polypeptides. We can use some receptors, other, the entities, and they specifically they can target, certain, tissues, right? The other one in the pegylated lipids. So pegylated is actually is known since Terry Allen from University of Alberta. She designed, let's make, pegylate the phospholipids, right? And since then, the area has really blossomed actually for the last 40 years. So the pegylated liposome, they came, through Terry Allen from University of Alberta, Edmonton. And you can see in every product, not every, but every, everything in liposome is pegylated. And the reason being pegylated, because they circulate longer, they keep circulating without being opsonized, being, caught by the, macrophages, right? Because otherwise end up in macrophages, and they'll be just, gone, then you'll be cleared by the EPR mechanism, right? And then last but not least, also the lipoplexus. When I started my career in liposome back in early 90s, we are very, very actively actually pursuing gene therapy, right? We published several papers, several patents, filed several patents. And then a few years later, the gene delivery was stopped because there was an accident. Actually, an incident happened in UPenn. A person died, so everything was put on the fully stop. But then it revived again in COVID vaccines, right? So back in the days, we are using some novel excipients, and the FDA, of course, it was very, careful not to approve because you need to have safety data to prove. Otherwise, it's safe. Otherwise, the drug will not approve. So back in the days, when we look in the DOTAP and other, the cationic lipids to stabilize the nucleic acids, right? So, but today, fast forward, actually, many years later, now the whole industry is looking into, more in the lipoplexus and, looking at the messenger RNA. And the first drug actually was approved on a battery. Right? When a pattern was launched, as a small interference, the RNA, and that was launched by Annalino. And then many years later, then the two vaccines came, COVID vaccines, because of the emergency use, right? And those, and that's really the challenge, the safety concern, because FDA was always in the long run looking for the safety data for the non-ionizable lipids, how safe they are before, given to the human, right? I'll come back. Then we also we have the cubosomes. There's a lot of interest in the cubosome because cubosomes are non-laminar structures, and they also have increased, helped more drugs in the drug loading. So all these structures, they have very limited space, to put the drug into the aggregates, also the lipid aggregates. But some of the aggregates are also better than the others and more stable than others, and therefore they are more widely used. But again, there is no product. There's a lot of papers. If you look into literature, they fill with the cubosomes, right? There are a lot of. So we have also written a paper as well on cubosomes. So if you're interested, we can share it with you. So just give an example. This is an example of a very BCS class 2 drug. Without going into too much in detail, so the challenge was actually how to stabilize, and also there's no drug actually. This is in a tablet, but it's not in the liquid formulation that on the liposome. So we designed actually. So that was our the challenge, and then we our task was to develop the injectable nanoparticle formulations, and also stabilize the drug molecules to achieve the desired sustained release effect, right? So these are, so let's say, that was our objective. And then able to, prepare, 3 different formulations. For any formulation, we try to do at least 3 or 4, 3 to 6 formulations for a client. So make sure that, if one is working, the other, if it doesn't work, that's fine. So because they have every formulation design, it has to go for the toxic studies, just to look at the efficacy of the drug. Otherwise, there's no use of, making, right? And then able with this way we achieve the right PK profile. We had a drug was released. And so we filed the patent as well. And so you look at the other one, the vaccines, for example. And this one actually already spoke earlier. The non-ionizable lipids. Actually, there are 4 lipids, right, in all these formulations. Also, people have a look at the different, why not 5, right? So there are some papers as well. And there's a lot of work going, Mansoor Amiji and others, they have done a lot of work actually on the messenger RNA, right. So if you look at the structure of this one, these are all different. Okay, I have 5 minutes to go. So I will just, go quickly. But the key is the message is actually how to really design or use a smarter, ionizable lipids. And there's, Bob Langer's and others, they have published a lot of papers. In fact, we also have, some papers as well on lipid salt, particularly the lipid nanoparticles. So, if you're interested, we can share with you. But the key message actually, because these all ionizable lipids, they have 2 different functions. One, they stabilize the nucleic acid because they're cationic in nature. They have the amino group. And 2, they also, when they get into the cell, they go through the endosomal escape. And then they create, they direct the cells to, they create the spike, proteins, and then spike, creates the antibodies, right? So that's where the mechanism, so how the antibodies really form, to the spike protein. So this is one example I'm giving. We have also formed a team with the alliance with another company. They are also, provide the different ionizable lipid, and so we have, we have some data, and it's very comparable to the non-ionizable lipid. So we have the, so if you're interested, we can provide some more data. So. And nanosol, I'll go to quickly actually. So we have several methods actually to prepare the nanosuspensions. We can go to the top, and down, and also the bottom approach. And wet milling is widely used, because people really want to grind it, right, to the maximum. So make sure you have highest surface energy. But the question is how you can, stabilize those particles. So they are, so there's a pros and cons, and and you want to high drug loading, right? So hide the, smaller particles, high drug loading, make sure the particles are stable, and also the, and they give the right efficacy. So we have all this technology. You can make nanosuspensions, and you can see these are big machines we have, and we are the only actually few CDMOs, maybe only one, that we have this delta beta. The gamma beta machines, right? So we have this in the ISO 5 room, and we can go up to all the way for 100 liters, right? With this machine, so we can do this, wet milling, with top-down approach, and then we can take it all the way to the very, mega scale as well. One example of PLGA. This is actually, I know some, Dr. Feng Zhang, he presented about the PLGA. PLGA is widely used in the, the long-acting injectable. Very recently, we had also webinar, and we invited Diane Burgess from University of Connecticut. She gave very eloquent, webinar. So we have also slides and also the webinar available, recorded. So if some of you are interested, we can share with you. But very simple, actually. The approach was, because we wanted to. It's a polypeptide. Wanted to control the Cmax, and also the extend the Tmax as well. They reduce, increase the half-life, and using this PLG approach, able to control, minimize the the Cmax, increase the safety profile, and also able to see the desired results, in vivo results as well. So just to summarize, the Ascendia has all the capabilities, both in the oral and also injectable space. And we can use, across the, all different modalities, different molecules. And so we can, and you can use both top-down and bottom-up approach, both for the. With that, actually, I would like to, I know there's no time for question, but actually we believe in, best practices, which is the brilliant technology, excellent services, superior quality, and also the trust. And please stop by to our booth. We have some of our colleagues here, so please try to meet with them. Dr. Mac is here, Matt Mackey, and also Bob Loader is here as well. And yeah, some of you interested, please stop by and we can talk more about it. Okay? And thank you for your attention.